About the types of immune response and antibodies. Now, the immune responses evoked on the antigenic exposure, these are of two types. First is the primary immune response and second one is the secondary immune response. So what is primary response? It shows the first contact of the immune system with the infectious agent, whereas all the subsequent contacts with the same pathogen, they present with the secondary immune response. As you can see in this figure, that the primary immune response, it is very short-lived. Okay. And it lasts for a smaller duration, we can say, whereas the secondary immune response, it is very powerful, prolonged, and it lasts for a longer duration. Antibody IgG, it is involved in the secondary response, whereas IgM, it is involved in the primary immune response. So we had said the term antibodies. What are these? Antibodies, also called as immunoglobulins. So these are the Y-shaped glycoproteins, as you can see in this figure, produced by the plasma cells on the antigenic attack. So the basic functional unit of each antibody, it is monomeric immunoglobulin. When two monomeric come, they form dimeric unit, and 5 they form pentameric. Okay. So in this figure, you can see that there are two chains. Okay, heavy chains and the light chains. Okay, two heavy chains and two light chains. And these are joined with the help of disulfide bridges. And in each heavy chain, two regions are there, variable region and the constant region. These are the types of immunoglobulins. First one is IgG. It is the most abundant immunoglobulin and it helps in complement fixation, protection of body fluids, and it can cross placenta. It is a very important point to memorize. Then IgA, it is a secretory immunoglobulin, so it is present in the body secretion. Largest immunoglobulin it is IgM. It cannot cross the placenta. IgE, it is involved in the allergic reactions, and IgD, it is having antigenic recognition property. Here the functions are enlisted again. Neutralization of the antigen or the attackers, helpful in the complement system activation. Agglutination, because immunoglobulins, they tend to adhere to the foreign antigens together in clumps which attract the phagocytes. They help in precipitation. Serum soluble antigens are glued together so that they can be precipitated out of the solution in form of clumps, which can be attractive for the phagocytic cells. They confer natural passive immunity also, and they have a role in hypersensitivity reaction, especially IgG, IgM, they are involved in type 2, type 3 hypersensitivity reaction, and IgE involved in the type 1 hypersensitivity reaction. Thank you.